This video is Chapter 5 Notes, Part 2. Facilitated diffusion. To remind you, diffusion is when particles spread out from high to low concentration. During facilitated diffusion, that also happens. Particles move from where there's more of them toward where there is less of them. However, the difference between diffusion and facilitated diffusion is that facilitated diffusion uses a protein channel. Normal diffusion would allow particles to just squeeze through the phospholipids that make up the phospholipid bilayer. However, those phospholipids can only move left and right so far. So if a particle is too big, those phospholipids might not be able to squish over far enough to let that big particle through. Also, this whole inner region of the phospholipid bilayer is very hydrophobic, meaning any watery molecule, or actually any particle that has a charge, cannot get through there. What would happen is the particle would start to move in, and the little phospholipid tail would kick that particle back out. So if a particle is charged or too big, it needs a protein channel to go through. That's what's shown in both of these diagrams. Still, concentration moves from high to low. The only difference is there's a channel for which the molecule moves through. On the contrary, active transport is not diffusion. Active transport requires energy. So active transport requires energy in the form of ATP. In active transport, particles are pumped against the concentration gradient, meaning against the way that they would normally flow. Shown here in this picture, we have hydrogen ions being pumped all to one side of the membrane. That is requiring energy. The reason is because particles don't want to be by those that are similar to them. So the natural flow of things would be for particles to spread out and be equal concentration on both sides. But in active transport, energy is being used to pump particles where the cell wants them to go. We saw that in both cellular respiration and photosynthesis, where a small amount of energy from the electron transport chain was used to pump hydrogen ions all to one side of the membrane. Same idea. Energy is required, so that is active transport. In conclusion, there's 3 ways in which particles can move across the cell membrane. Number 1 is diffusion. Particles spread out from where there's a lot to where there's less. They squeeze through the phospholipids in the bilayer. Method number 2, facilitated diffusion. Again, moving from high to low concentration. Difference, we need a protein channel because these particles are too big or too charged. The third, active transport, requires energy in the form of ATP. Particles are moving from low to high concentration, which is not the way that they would naturally go. So the cell is expending energy to move a particle where it wants that particle to go. There's 2 big examples of active transport, which are endo and exocytosis, moving huge molecules like huge chunks of food or huge amounts of waste out. So we've got the movement of huge stuff. Endo and exocytosis. It's endo means to enter, exo means to exit. So in endocytosis, the cell membrane is pinching in to allow a big molecule into the cell. And in exocytosis, the cell membrane is pinching outwards to release a huge particle. You don't need to know a ton about those, but it's possible that you could see them in the future. To conclude this video is water potential. Water potential is kind of a beast at first, but hopefully you'll see that it's not too crazy. It is a math formula. So kind of like Hardy-Weinberg, this is one of our few math formulas of the year. In water potential, we of course are talking about water. So water potential goes with the idea of osmosis. And we are always going to be talking about the flow of water via osmosis being passive, where water moves from high to low concentration. Water moves from where there is a lot of water to where there is less water. We call that the concentration gradient. So water potential is a math formula. It is a math formula that uses the symbol psi. Kind of looks like a trident from like Little Mermaid King, but it is a Greek letter psi, and it is made of pressure potential and solute potential. So this guy here is water potential. And it's made of pressure potential and solute potential. What these psi's represent are numbers. So try not to get confused by the numbers. Essentially, a high psi, so a high number, indicates a lot of water. And a low number represents less water, aka more solute. So when I plug numbers in, I would have a water potential for inside the cell, and I would have a water potential for outside of the cell, and I would compare those numbers to figure out which way water is flowing. And water would always flow towards where there's less water, so it will always flow toward the lower value. So, for example, if I have a cell here, and let's say my water potential of the cell is -20. I'll come back to how we get these numbers in a second. But if the water potential is -20, and they tend to be negative numbers, and my environment has a water potential of -5, I would figure out where is the higher number. Which value indicates more water? If I compare -20 and -5, my lower number is more negative, so my higher number is -5. Water will flow from high to low. In that example, water would flow into the cell. That indicates to me a hypotonic environment. The opposite would then be true for a cell in a hypertonic environment, where the water potential of the cell would be lower than the water potential of the environment. In which case, water flows from high to low. -2 is a higher number than -10, so that would indicate a loss of water. All right, so how do we get these numbers? These numbers come from a math formula. So to start, we are looking here at this formula, water potential. It's made of two things, pressure potential and solute potential. So first up is the easier one, pressure potential. Pressure potential will always be 0 unless you are told otherwise. What pressure potential is, it has to do with turgor pressure. Turgor pressure is an idea that we see in plant cells where the plant cell wall pushes back to prevent continual uptake of water. Now, if we think about this, if I have a plant cell and I am going to give it pure water, by definition of osmosis, water should continue to flow into that cell because I'm watering my plant with pure water, which is what we always pretty much do. We water plants with just plain water or the rainwater. So then water should continue to flow into that cell and in theory get bigger and bigger and bigger and bigger and then burst. However, we don't see that. We don't see that because plant cells have this thing called turgor pressure, which is based on the cell wall of the plant that goes around the outside of the membrane that pushes back on the cell saying, stop taking in water. That then prevents the cell from bursting because it's this opposite force where water is flowing in, the wall is pushing back saying stop taking in, so eventually the plant does stop taking in water. It does not end up bursting. In a lot of osmosis scenarios, we are talking about animal cells, and in animal cells there is no turgor pressure, which is why we normally assume that the value is 0, unless a question specifically says, in this scenario, the pressure potential is 1, or something along those lines. So we can usually just ignore pressure potential. The other piece is solute potential. Solute potential, we never ignore. Solute potential is the big guy. So solute potential is a formula, negative I CRT. The unit on solute potential is bars, like how I would measure 4 centimeters, 4 grams, 4 bars. So the unit is bars. Because of that negative sign, these values, like we saw above, are almost always all negative. All right, so we have 4 variables here, I, C, R, and T. I'm gonna go through those. So first up is I. I is representative of the ionization constant. When I put a solute, when something gets dissolved in water, that ionization constant will either be 1 or it will be 2. It will be 1 for sugar, because when I dissolve sugar in water, the molecular shape of sugar stays the same. It will be 2 for salt, because when I dissolve salt in water, salt breaks apart. Like NaCl would break into sodium ions and chlorine ions. So that's where I get the 2 from, because it was 1 molecule that breaks into 2 pieces. C represents the concentration of my sugar or salt, meaning like the number of moles of salt or sugar that's dissolved in the water. R is a constant, meaning R is always going to be the same value. And that value is 0.0831. It's always the same. The unit on that is liters bar per moles of Kelvin, but 0.0831. And then T represents temperature, and it is temperature in Kelvin, which to find, you do the degrees Celsius plus 273. So this formula, negative ICRT, is how we can find the values to determine which way is water going to flow. So this whole idea of water potential takes something very simple, water flows from more to less, and makes it much more complicated. So we'll do an example question together. I just gave myself more room to write here that I didn't actually need. So here we go, an example. What is the water potential of a 0.5 molar sucrose solution at 25 degrees Celsius? All right, here we go. I know water potential is made of pressure potential plus solute potential. I see nothing here about pressure potential, so I know that that value will be 0. I now need to find my solute potential, negative ICRT. This example is sucrose. Anything that ends in ose is a sugar. My sugar does not split apart, so my I value will be 1. Next is C. C is concentration in moles. Here it is, 0.5 molars. R is a constant. It is always going to be 0.0831, always. And then lastly is temp. To find temp, I will do my degrees Celsius, 25 degrees Celsius, plus 273. And I'm now ready to grab my calculator and solve. What I get for that is going to be negative 12.4. When I add that to 0, 0 plus negative 12.4 will give me an answer of negative 12.4. The unit on that is bars. That alone does not tell me anything about which way water will flow. So I would need a second value to which to compare that number to figure out which of those two is higher, and thus that higher one is the one that would lose water.